Hello, this is Professor Matt Katroulis with the next Chem 110 video lecture. This is experiment number 16 on aldehydes and ketones, which continues our study of organic chemistry. This experiment is divided into three parts. The first two parts involve primarily looking at structures and looking at properties of different compounds. So for part A, we're going to be looking at some models of some different aldehydes and ketones. Now, aldehydes and ketones both contain the carbon-oxygen double bond. So do many other functional groups, including carboxylic acids, esters, amides, and some others. But we're going to be focusing primarily on aldehydes and ketones because in some ways they're similar in structure and reactivity. Now, we call the carbon-oxygen double bond a carbonyl group. Now, just because we call it that doesn't mean that you should use that term for functional groups. The word aldehyde and ketone is the word you should use when you're talking about functional groups. With aldehydes, that carbonyl group is bonded to at least one hydrogen atom. In the case of the compound called formaldehyde, it's actually bonded to two different hydrogen atoms. But that is unique. That is the only compound where you have an aldehyde with two hydrogens. All the rest have a carbon bonded to only one hydrogen. In ketones, there is no bond between carbon and hydrogen on the carbonyl. Instead, that carbon is going to be bonded only to other carbons. So, for example, it might be bonded to a methyl group and a methyl group, or it might be bonded to a methyl and an ethyl group, or it might be even more complicated than that. Now, as I show you these models, what I would like you to focus on is the shape and the bond angles around that carbon of the carbonyl group, the CO double bond. Think about this for a second. The carbon is making a double bond to oxygen, so it's bonded to an oxygen atom, and then it's bonded to either a hydrogen and a carbon, in the case of an aldehyde, so that's three different atoms, or it's bonded to another carbon and another carbon. So again, that would be a total of three different atoms. So what would the shape be for an atom bonded to three other atoms with no lone pairs on that atom? If you'll recall, that was called the trigonal planar shape. And the bond angles around a trigonal planar atom should be 120 degrees. So definitely pay attention to that. As for naming, I'm not going to go over naming in this lab video because I cover it extensively in my online video for Chapter 12, the one that is on aldehydes and ketones. So I'm going to go ahead and put a link to that down below this video, and I encourage you to go ahead and take a look at that if you're running into difficulties. Now, one of the problems with the lab report sheets is that in order to save space, they sometimes compress your answer boxes far too much. So I've come up with another page that I would prefer you use instead of page 183 to turn it in. Basically, what I've done here is I've just expanded parts A and B from one page with little boxes to two pages, one for part A and one for part B, with a lot bigger boxes. So I would strongly recommend that instead of turning in page 183, that you turn in those two pages in the link that you'll see below. If for some reason you don't have access to a printer, you're more than welcome to draw it out on two separate sheets of blank paper instead and submit those. Here's our first molecule, the simplest of the aldehydes, formaldehyde. It is the only aldehyde in which carbon has hydrogens on both sides. All other aldehydes contain at least one bond between the carbonyl carbon and some other carbon atom. This compound is acetaldehyde. That is the common name, just like formaldehyde was the common name of the previous compound. Here we've added yet one more carbon to the chain, and this compound is called propionaldehyde. That is the common name. Here is our first ketone. It is the compound called acetone. Acetone is, again, a common name. This compound is called butanone or 2-butanone. In either of those cases, those are both the IUPAC name. You are asked to find the common name on your lab report sheet. That's pretty easy to do. You can go ahead and Google butanone and you'll find that. I'll give you a hint. Its abbreviations are MEK. And finally, this is cyclohexanone. Notice the 6-membered ring. This is not a benzene ring. Benzene rings are flat, which this is not. Cyclohexanone is the IUPAC name. There is no common name. We now move to Part B, which looks at the properties of various aldehydes and ketones, including some ketones and aldehydes which will appear in Parts C and D, and others which will not. The instructions tell you to look up the structures of each of the molecules either on the internet or by looking up their structures in a manual. I'm going to instead provide models for you to look at, just like I did in Part A. Now, the first section asks you to take some of these samples and smell them, and some of them have a very pleasant smell. But unfortunately, we have not yet developed the technology to allow you to smell over the internet. Whether that's fortunate or not, I don't know, but for the purposes of this lab, that's not going to be possible. So you can look up what each of these compounds should smell like on the internet, and in many of the cases, the name should be pretty obvious as to what it smells like. For example, cinnamaldehyde should give you a big hint as to how that compound smells. The only one of these that might prove to be a bit difficult for determining the smell is camphor. And the words that I've seen used on the internet to describe its smell are minty or medicinal. And medicinal can mean a lot of different things to people. When I see medicinal, I usually think it smells like rubbing alcohol. And camphor doesn't really smell anything like rubbing alcohol. So I'm going to just tell you this, and that is if you've ever applied Vicks Vapor Rub to your chest when you've had a cold or a fever, that's the closest thing I can come to in terms of describing the smell of camphor. It's like menthol, which is in Vicks Vapor Rub. It is also somewhat similar to eucalyptus, particularly if you take eucalyptus leaves and kind of shred them. That is a fairly close analogy to the camphor smell. Now, in the next column, for Part B, it will ask you to draw out condensed structural formulas, and I would prefer that you not draw out the hydrogen atoms on any rings, so that would include cyclohexane or cyclopentane rings or benzene rings. It's understood by chemists that when we see those rings, that each point on the ring stands for a carbon with the appropriate number of hydrogens. So for example, if I were to ask you to draw 3-methyl cyclopentanone, that's going to be what? Cyclopentanone would be a 5-membered ring with one of those carbons having a ketone, and then 2 carbons away on carbon number 3 you would have a methyl group. Look at the drawings I give you here. The one on the left is more closely defined as a condensed structural formula, which is what you're asked for. And so it's an acceptable answer, but it's a bit messy and cluttered. I prefer that you show carbons in rings just as points. And then you can go ahead and expand any substituents that are bonded to that ring. And then in the final column of Part B, you're asked to identify the compound as either an aldehyde or a ketone. And you just write one word or the other. So for example, in this particular case, we have a carbon double bonded to oxygen. We have a carbon on this side and a carbon on that side, so this is a ketone. I'm going to show you some models now. I'm not going to show you the model for acetone because we already looked at that. So here is the model for benzaldehyde. Notice the benzene ring represented as the flat 6-carbon ring with the dotted lines which represent the alternating double bonds. Now rather than show you a model for camphor, I'm just going to show you its structure here. If you'll notice right here, you'll see that we have a 6-membered ring, like a cyclohexane ring on the bottom. If we look, there is what we call a carbon bridge going across from one side of the ring to the other. And then on that top of the bridge are 2 methyl groups. There's a methyl group where the ring joins, and there's a ketone. That's called a bicyclic compound. Here is vanillin. You'll notice the vanillin has a benzene ring. And there are 3 different groups attached. An aldehyde. And then we have an OH right there. So that would be an alcohol, but it's on a benzene ring. And what do we call it when we have OH groups on a benzene ring? Those are called phenols. And then finally at the bottom we have an oxygen. And that oxygen is bonded to a methyl group. So what functional group is that? Carbon oxygen to carbon. That is an ether. Let's spin it there to give you a better look at vanillin. And the final compound we're going to look at is cinnamaldehyde. Cinnamaldehyde also has a benzene ring, as we see. And attached to that is a carbon-carbon double bond. And attached to that is an aldehyde. Now there are 2 actual different compounds called cinnamaldehyde. There is cis-cinnamaldehyde and trans-cinnamaldehyde, depending on the carbon-carbon double bond. Which one is this? Look at the carbon-carbon double bond. Are the hydrogens pointing to the same side or different? They're pointing to opposite sides, so this is technically trans-cinnamaldehyde. In Part C, we're going to look at the solubility of some aldehydes or ketones in water or aqueous solution, as well as 2 chemical tests, the Iodoform test and Benedict's test. So normally the way we would test solubility is we would take a few drops of the different compounds that we're going to test and add them to a small amount of water and see whether or not they mix. And since all of these compounds are liquids, what we're going to be looking for is whether or not they form 2 different layers with water. If they form different layers, then that means they do not mix and that they are not soluble. If we were testing solids, we would expect the solids to simply remain solids and most likely stay at the bottom of the test tubes. So what do we expect in terms of solubility of these compounds? Well, to figure that out, we look at the intermolecular forces. Aldehydes and ketones have a carbonyl, which has an oxygen, which has lone pairs on it. Now, those oxygens are able to accept hydrogens in hydrogen bonding. So we might expect them to be soluble in water, but there is a limit to that. As the number of carbons increases on the compound, we would expect them to be less and less likely to be soluble in water. So, for example, compounds with about 4 to maybe 5 carbons are probably going to be soluble in water. As you exceed 5, it becomes very much less likely that they are going to be soluble in water. So, rather than do a separate solubility test here, I'm going to combine this with the iodoform test, because the iodoform test will require us to combine our ketone or aldehyde in a solution which contains water. And furthermore, that's an iodine solution, so there'll be a very, very big color indication showing off whether or not we have 2 layers. So it's going to be easier to figure out whether or not they're soluble by just looking at the end of the video I'm going to show you for the iodoform test. So let's talk about the iodoform test. It is specific for identifying not just ketones, but only a certain type of ketone called a methyl ketone. A methyl ketone has at least one of the carbonyl groups occupied by a methyl group. So let's see what I mean by that. Here are 3 different compounds with carbonyl groups. Carbonyl again means carbon double bonded to oxygen. Look at the first compound. We have a carbon bonded to a carbon on the left and a carbon on the right, so that makes this a ketone. On the left side is a methyl group. So therefore, it is not just a ketone, it is a methyl ketone. So we would expect this to give a positive test for the iodoform test. So I'll tell you more about what that means, you'll see in a second. But it is a methyl ketone. This next compound, which is 3-pentanone, has a ketone, carbon double bonded to oxygen. That carbon is bonded to a carbon here and a carbon here. This side is an ethyl group, and this side is also an ethyl group. So neither of these are methyls. So even though it is a ketone, it is not a methyl ketone. Therefore, we would expect a negative iodoform test because it's not a methyl ketone. And this compound right here has a carbon double bonded to oxygen, but it's bonded to hydrogen. So this is an aldehyde. So it doesn't even matter what this side is. Generally, we would expect this to not give a positive iodoform test. Now, technically, for reasons that we're not going to get into, if this was a methyl group, it very well might give a positive test, even though it's an aldehyde. So there are some exceptions to these tests, but we won't see any of those exceptions in what we're going to look at in our experiment. So if we take an iodine test solution, and that contains iodine, I2, and potassium iodide, Ki, and we add it to a methyl ketone, and then we take that methyl ketone and make the solution basic by adding sodium hydroxide, we will get a yellow solid called iodoform, CHI3, if it is in fact a methyl ketone that we're adding. If we're adding almost any aldehyde and most ketones which are not methyl ketones, then we would not expect to see that yellow solid. So methyl ketones will give you a yellow solid iodoform under these conditions. Now, I've tested out the procedure along with my colleagues, and I have not found it to be a very good procedure for producing the iodoform test. So I'm going to change it. And so the way I've changed it is the following. I'm taking an equal amount of the aldehyde or ketone that we're testing, in this case 20 drops, and adding it to the same amount of the iodine solution, which has iodine and potassium iodide. Now the iodine solution is brown. The compounds we're testing are clear and colorless. And if we're taking something clear and colorless and adding something brown to them that doesn't initially react, that turns everything brown. I then add dropwise 10% sodium hydroxide solution, and that will cause the brown color to start to disappear. So I add just enough drops to make the brown color disappear. Some people would recommend at this point adding one more drop of iodine, and that's something I might recommend testing, but it's not what we'll see in this video. And then what I'm going to do is heat the test tube for several minutes at about 70 to 80 degrees Celsius. The lab manual says 60 to 70, but I found 70 to 80 to work better for this experiment. So without further ado, let's go ahead and take a look at the video that goes along with this experiment. Here you see me adding the iodine reagent to the first of the aldehyde or ketone solutions. This first one on the left is propanal. Notice that the propanal, like all the other ketones or aldehydes that we'll be testing, is clear and colorless. And notice that the iodine reagent is brown. Now, rather than showing my adding the solution to every one of the test tubes, I'm just going to show it for propanal. Here you see the test tubes after the 20 drops of each ketone or aldehyde have been mixed with 20 drops of the iodine solution. Now, pay careful attention to the cyclohexanone, and you notice that the cyclohexanone clearly exists in 2 layers. And that is because cyclohexanone is not miscible with water. Recall that the iodine solution is mostly water, so that split into 2 layers indicates that it's not soluble. Here you see me adding just enough of the sodium hydroxide solution to get most of the brown color of the iodine to dissolve. And I have to mix that to keep separate layers from forming. And still there is some, but there's enough mixing for there not to be a problem. And then I'm going to take each of these solutions and put them in a hot water bath once I've accomplished this. Here you see the 4 solutions. On the left is propanal. Next, to the right of it, is the benzaldehyde solution. Then we see the acetone with a clear yellow precipitate in the bottom of it. And then furthest to the right is the cyclohexanone. You'll notice that after some time, unfortunately, the benzaldehyde will start to boil off. And you see that it's not as soluble as we had anticipated. And in fact, you could say that the benzaldehyde is essentially insoluble with water. Here we see propanal at the end of the iodoform test. No yellow precipitate. Benzaldehyde. No yellow precipitate. I'm going to shake that a bit. Acetone does seem to have a yellow precipitate at the bottom. Now, cyclohexanone might appear to have a precipitate, but let's go ahead and look at that from the bottom. You'll notice, if we look very closely, that there aren't really any crystals. Oops, let's see if we can get the camera focused. There we go. All there are liquid droplets that are mixed in with the solution. So there's no precipitate with the cyclohexanone. The acetone, on the other hand, you can clearly see solid precipitate at the bottom of the test tube. So the conclusion here would be that the only one of these which gives a positive test is acetone. And we also notice that the benzaldehyde and the cyclohexanone are not soluble in water. In this final part of the experiment, we will be exposing the 4 different aldehydes or ketones to Benedict's reagent. Benedict's reagent, which contains the blue copper 2 ion, is able to oxidize aldehydes. That's something that we learned when we initially were studying alcohols. Primary alcohols can be oxidized to aldehydes. Aldehydes can be oxidized into carboxylic acids. We also saw that secondary alcohols could be oxidized into ketones, but ketones cannot be further oxidized. And so if we combine ketones with Benedict's reagent, we would expect nothing to happen. And so it is useful for some circumstances between telling apart aldehydes and ketones, although in my experience this is a not very consistently reliable test except for very specific types of compounds, namely carbohydrates, which we will be discussing in an upcoming lecture. So Benedict's reagent initially becomes green during a positive test, and then it will completely go into becoming the red precipitate copper 1 oxide given enough time and the right conditions. So we should take a solution remaining blue as a negative test. If it becomes green, that should indicate a positive test, and then it can go from there into being the red copper 1 oxide. Now, in some cases, you might end up with a mix of green solution with the copper 1 oxide dissolved in it, and you might only see the green in those particular kinds of circumstances, and that should still be taken as a positive test. Really, we should only take it if it remains blue as being a negative test.
Now I'm going to do Benedict's test. So I've added 10 mLs of each ketone and or aldehyde to a test tube. I have now added 2 mLs of Benedict's reagent to each of the test tubes. The test tubes are placed in a boiling hot water bath. And here we have the conclusion of the test with Benedict's solution. So we can clearly see that the cyclohexanone and the acetone still have the same blue color as Benedict's. However, propanol and benzaldehyde have gone to a greenish, indicating some oxidation has occurred.